Interactive and visualization-based tracking in FluoRender. This talk is about the latest development of FluoRender. Recently, we added a series of features into FluoRender and would like to make a versatile tracking system for biological research. This is because the capability to track cells and other organelles from microscopy scans plays an important role in understanding the functions of biological structures, their development, and relations to diseases. FluoRender is an interactive tool for fluorescence microscopy data visualization and analysis. It already integrates a series of modules. Multi-channel visualization allows a large number of volume channels to be rendered in an anatomical atlas. The interactive tools allow users to select and segment structures directly in 3D. The 4D visualization features allow viewing time-dependent scans at interactive speed. We can also automatically segment structures, such as cells, from a 4D dataset. All these features are implemented taking advantage of the processing power of GPUs. It is only natural to bring all these functions together for an intuitive and versatile cell tracking system. Cell tracking is a research area that has been investigated by many groups for a long time. There have been a plethora of algorithms and tools for cell tracking. Here, we refer to the surveys by May-Jering et al., which listed different cell tracking methods and grouped them into categories. More recently, there have been studies on the performance of different methods through competitions. The comparisons of the cell tracking results using standard benchmark data were discussed in the work of Maska et al. And Chanuard et al. Although a thorough comparison of these methods is not our research goal, we were able to experiment with several recent cell tracking tools and algorithms. For example, Mosig et al. And Chatterjee et al. Used the maximum graph matching for cell tracking, and Amat et al. Used the probability association methods in their cell tracking tools. However, the benchmark data used to test these tracking methods are usually more simple and clear than data acquired from real-world research. For example, existing tools are not robust for densely packed cells. Here is an example of using the tool by Amat et al. To track cells in a confocal microscopy scan of zebrafish eye development. We colored those cells that were tracked by the tool. We can see that the tool was able to track only a small number of cells. Many of the colored cells were also incorrectly segmented or tracked. The tool provided several parameters for users to tweak. This was actually the best result we could obtain after exploring all possible parameter combinations. Another method we examined was the maximum bipartite matching. As illustrated in this example, cells from two time points are organized as vertices on a graph. The graph is bipartite because there is one group of vertices for each time point and only cells from different groups can be matched. The matching of two cells is represented by an edge on the graph. Edge weights can be assigned based on the spatial relationships of the cells. The solution of the cell tracking problem becomes finding the matching that maximize the edge weights. This method is not robust because segmentation errors can influence the matching results. Here, cell B is over-segmented. The result from maximum matching not only mismatched B'2, which is part of B', but also got the tracking wrong for C and D. We found that, when we applied these tools and methods to real-world cell tracking problems, the tracking accuracies were often lower than their published results. We do not doubt the integrity of the previous research, but still we would like to ask for the reason, or reasons, why these methods didn't work for real-world research. One could actually give many reasons, such as that the real-world data we used were too much complex, that users of the tools lacked the experience, the parameter settings were not intuitive to use, or it could be just the Murphy's Law. But we think there are two more fundamental reasons for previous tools' problems. The first is the system design, and the second is the method for validation, or evaluation of the tracking results. Let's look at the system design of previous tracking tools. Most cell tracking tools were designed as a linear process. In this example, the cell tracking tool has 3 steps: image enhancement, cell segmentation, and cell linking. From a point of view of system design, the linear systems are usually not robust because of the lack of quality control. It needs feedback among the modules to make sure the results are correct. Errors can easily propagate from one step of the processing pipeline to the next. This is especially a problem for time-dependent data, because error may propagate from one time point to another. This design may be also limited by the choice of algorithms. For each step, only one algorithm can be used at a time. One may say that we could choose the best algorithm, for example, for segmenting cells. But in reality, different algorithms are usually balanced by their advantages and shortcomings. The linear design doesn't allow different algorithms to work together. Finally, most tools lack the intuitive user interactions to diagnose and correct errors in tracking. Let's look at the problems of validation for cell tracking next. Conventional cell tracking results are validated against benchmark data. The ground truth of the benchmark data are established by either manual work or simulation for synthetic data. The tracking result of a tool is then compared with the ground truth to determine if the tool is effective. Different tools can be also compared against each other using the same benchmark data set. The first problem is that the benchmark data can't cover all the features from real-world scans. Innovations in the imaging techniques are bringing new scans every minute. A tool works for a benchmark dataset doesn't mean the tool also works for a new dataset. When the results of a tracking tool for a new type of data can't be validated from the old benchmark data, we need to redo the benchmarking. However, manual work is the only method available. For large and complex 4D data, it is simply impossible to generate the ground truth by manual work. Also, if we can generate the ground truth every time, there is no need for an automatic tracking tool. Another problem is that, once a tracking tool fails for one dataset, or part of a dataset, it is very difficult to locate and diagnose its issues and fix them. This is also done manually in previous research. If one needs to manually check and fix all the tracking issues, there is again no need for an automatic tracking tool. Since we identified the two fundamental problems in cell tracking as system design and validation, our work has been to address the issues. It leads to the two parts of this talk. The first part is a description of the system design for cell tracking in FluoRender. We adopted a nonlinear and iterative design. Different modules of the system include our previous and ongoing work. The second part presents a novel validation method for cell tracking algorithms. We call the method the uncertainty footprint. This part of the talk will also be presented at Eurovis later this year. Considering the issues in system design of previous cell tracking tools, here are the design objectives for our system. We want to include many different algorithms from image processing research in our system. We want to have intuitive manual operations for result correction, which will take advantage of existing visualization techniques in FluoRender. Finally, we want that different methods, including manual operations, can work together. The basic idea for our system design is quite simple. We add a loop into the cell tracking workflow. The 4D data are first properly processed and segmented. The segmented cells are linked using a graph matching method. We will detect problems in graph matching. Because many of the matching problems can be caused by segmentation errors, we resegment the regions that cause the matching issues. Then, we detect problems from resegmentation and ask the user to manually correct them. The manually corrected results are sent back to graph matching again. The system will be able to learn from the user inputs and fix issues automatically. This design allows feedback from one module to another. Based on the feedback, different algorithms can be chosen to best deal with specific problems. Let's look at individual modules of our system. For initial segmentation, we use the synthetic Brainbows. It is a GPU-based framework for assigning unique IDs to structures in a volume. We have made several improvements to the original framework. It includes multiple criteria for separating structures. It is suitable for quickly segmenting a 4D dataset and showing the segmented results immediately. The basic synthetic Brainbows processing has two steps. In the first step, we randomly assign integer IDs to the voxels of a volume dataset. And in the second step, we grow IDs similar to a diffusion process. The ID growth is controlled through a series of criteria, so that the final result can label different structures with different IDs. Choosing the criteria for ID growth is important to using the synthetic Brainbows framework for segmenting cells. Here are several examples of different criteria for ID growth. For disconnected structures, using the intensity value to stop ID growth would be sufficient to separate cells. When two cells are connected but the intensity is high at cell centers, we can use the gradient magnitude to stop ID growth and separate the cells. When cells are fused together and the gradient magnitude can't separate them, we can use a density criteria. This is because the place where they connect has lower density if we use a reasonably sized window. This is similar to the density-based methods for clustering. Finally, we can count the voxel numbers within each structure, and filter structures based on size. This allows us to remove noise easily. All of these criteria are implemented on GPUs for fast data processing. After the establishment of the initially segmented results, we use graphs to describe and store the spatial relationships of the components from initial segmentation. The segmented cells are vertices on a graph, and the graph edges represent the spatial relationships among cells. To track the cells, we would like to find the most likely matchings of all possible edges. Since the initially segmented results may not be 100% accurate, we may want to modify the segmentation and therefore the graph. We design the graphs so that its components can be dynamically modified in the matching process. Here is an illustration of the data structures for graph matching. For a given time point t, all segmented components, or cells, are organized by a graph. We call this graph the intraframe graph. Its edges describe the neighborhood information of the segmented cells. Since one cell may be over-segmented, having this neighborhood information is useful because, for example, the two parts of an over-segmented cell can be easily found and combined in the matching process. Considering that the components may be combined, we also build a list of nodes as parents for the components. Initially, each component is linked under one node. Once components are combined, this data structure will look like a tree of two levels. The same data organization is repeated for each time point of the sequence. Finally, to describe the spatial relationships between two time points, we build an interframe graph. The cell tracking process is to find the valid edges on the interframe graphs. We build our system to detect issues from the graph matching process. Many of the issues are related to incorrectly segmented cells. When the segmentation issues are detected in graph matching, we need to redo the segmentations for local regions. We use several commonly used clustering methods to handle both over and under segmentations. The most simple clustering method we use is the k-means. We also use a density-based method to segment connected structures. For shapes that can be modeled as Gaussian distributions, we use the EM method for Gaussian mixtures. Notice that cells cannot always be modeled as Gaussians. The first two methods don't rely on shape modeling. However, k-means and EM clustering also require the number of clusters. The system design will allow us to choose among these methods, depending on the feedback from the graph matching process. Since automated matching and segmentation may not handle every detail, we allow users to interactively view and fix the tracking issues. This is based on the paintbrush tools in FluoRender. To select a cell, we enable the paintbrush tool and use the mouse to paint on the visualized results. The brush stroke defines the selection region in the 2D space of the rendered image. The 2D selection region is projected into 3D space. Structures and voxels outside of the projected region are excluded from the selection. For the selection to follow the intensity variation of underlying data, we can perform an automatic threshold estimation. We first generate a histogram of all the voxels within the projected region, and then perform a histogram-based segmentation. Only high-intensity voxels are selected in the process. To prevent the selection of occluded structures, we send rays from all selected voxels back to the image plane. If a ray is occluded, the origin voxel of the ray is excluded from the selection, because it can't be seen by the user. Finally, we grow the selected voxels by iteratively evaluating a diffusion equation. The intended structure is then selected. We can use the paintbrush tool to select cells and perform a series of operations to modify the tracking results. When a cell is incorrectly linked to another cell, we can select both the cell and the correct one, and perform an exclusive link. It breaks all existing links and then links the selected ones. Sometimes we want to link more than one cell to a single cell. This is because cells may split or one cell is over-segmented. We can select the cells we want to link and perform a link operation. It won't break existing links. If we just want to unlink cells, we can select one cell from either end of the link, and then perform an unlink operation. To fix segmentation errors, we can select one part of a structure and assign a new ID. The structure with the new ID can be then linked to other structures. If an ID has already been assigned, the selected part will receive a new ID. It essentially separates the selected part from the original structure. Similarly, we can also merge two different IDs into one structure. Theoretically, we could use these operations to perform a purely manual tracking. We have talked about the individual components of our cell tracking system. However, a key component is missing from the system. That is how to detect issues from the results of one module, and then allow another module to fix these errors. Without a method to evaluate the results and detect issues, the loop in our system can't work. The method we propose is called the uncertainty footprint. Instead of looking at the resulting data directly, it analyzes the behavior of the algorithm itself and finds whether the algorithm is confident to process a data set. If the data processing is uncertain, it also points to regions with high uncertainty. We can use the uncertainty footprint to detect tracking issues and fix them. These are the research goals for the uncertainty footprint. We would like an evaluation method that doesn't require manual examinations. We want this method to be generally applicable, so that it can be used for different algorithms in cell tracking. We will use this method to detect tracking issues. Finally, it adds feedback to our tracking system, and we can use one module to fix the issues from another module. The uncertainty footprint will allow different algorithms to work together in our tracking system. Evaluation an algorithm based on its behavior during data processing is a less researched area, as most evaluations are done by comparing results. Here are some of the inspirations we got when we approached this problem in cell tracking. First, tracking is an optimization process that finds the most likely matching. Many optimization algorithms are iterative. For iterative algorithms, we usually accept or decline a result based on convergence. We examine if the sequence of results over the iterations converges on a stable solution. A converged result may not be correct, but it is what we usually will accept. The most commonly used convergence criterion is whether the difference between two outcomes of two iterations is sufficiently small. We wondered if we can get more information than that from a convergent process. For example, how confident, or uncertain, a process achieves its convergence. This information may shed light on the issues of the process if it is uncertain. We would like to dig into some background about the concept of convergence in mathematics. The mathematical definition of convergence was first studied by the renowned mathematician Augustin Louis Cauchy. He once proved and published a theorem about the continuity of function sequences. In his work, he stated that the limit of a convergent sequence of continuous functions is also continuous. The seemingly intuitive result was in fact erroneous. A young mathematician, Niels Henrik Abel, found several counterexamples. Here is one of them. If you don't recognize the equation immediately, what Abel proposed was the Fourier series of a sawtooth wave. The wave itself is linear within each period, and is continuous between two periods. It can be approached by the combination of a series of sine functions, which are continuous. Apparently, Cauchy was wrong about his theorem. But what's the problem? It took another generation of mathematicians to finally figure out the problem. And today we know that Cauchy's sequence has to converge uniformly in order to converge on a continuous function. On the other hand, it also tells us that non-uniform behavior can be easily observed in the convergence of functions. Specifically, a function can converge at different rates at different locations on its domain. Here is an example from wikipedia.org, which is much simpler than Abel's counterexample. The powers of a sine function are continuous until they converge to the Dirac delta function, which is discontinuous. We can see that the converging speed is getting slower and slower from two sides toward the center within the domain of 0 to pi. The difference of the converging speed essentially rips apart the function. We think the non-uniform behavior in convergence is related to the uncertainty in many optimization processes. To take advantage of this phenomenon, our first question is, can we visualize it? If you think the Wikipedia example already provides a visualization, what about functions in higher dimensions, as we often see in many optimization problems? Here is our solution to our questions. We first quantify the converging speed by aggregating the local value changes over the iterations. In the equation, fx is a function within a sequence. At each point x on its domain, we sum up all the differences between two iterations, which is our gx. The key concept here is that gx is a real-valued function sharing the domain with fx. It represents the total value changes at each point on the function domain. When gx is a real-valued function over a spatial domain, it is essentially a grayscale image, or volume. We use one of the most commonly used techniques to visualize gx, the histograms. There are several advantages of using histograms here. First, a histogram is not limited by the dimensions of gx. It can be applied to images, volumes, or higher dimension optimization problems. Second, we inherited many interactive and image processing methods from previous research. It allows us to study this problem easily. For an iterative process, we first generate a 1D histogram of aggregated value changes for each iteration. Then we put all the 1D histograms together to form a 2D histogram. Non-uniform behavior within the process is visualized as distinctive patterns on the 2D histogram. When a local value stabilizes, a horizontal line is shown on the histogram. Different regions of the domain may stabilize with different aggregated value changes. These are shown as horizontal lines with different heights. If a value doesn't converge, its diagonal line will continue. When lines separate, we know that divergent behavior might be isolated. It leads us to examine the areas with potential problems. We think that the uncertainty footprint is a generally applicable method. It can be used to study the behavior of iterative algorithms on spatial domains. Since we designed the uncertainty footprint for cell tracking, we will focus on its applications to cell tracking next. Earlier, we discussed the individual modules in our cell tracking system. Here, we look at two components of our system. One is for cell identification, or segmentation, and the second is for cell association, or linking. We will demonstrate the results of the uncertainty footprint with one algorithm from each. Specifically, we will examine the EM clustering algorithm for cell segmentation, and the graph matching algorithm for cell linking. The EM clustering method can be used to segment cells that are modeled with Gaussian functions. We are estimating a Gaussian mixture to maximize the likelihood of the mixture matching with the underlying voxel intensity distributions. We can see that the objective function of the optimization problem is high-dimensional. However, the uncertainty footprint can be easily computed and visualized, because we are accumulating the local likelihood changes for each voxel. Let's see several examples from real data. The first example, example A, has two cells fused together. We can see the shapes of the cells, but it would be difficult to separate them apart using intensity-based methods. We show that we started with two Gaussians well separated. The color-mapped aggregation values shows that the changes are concentrated on the border between the two cells. From the 2D histogram, we can see that the convergence was achieved easily. Example B uses the same cells from the first example. The difference is that we changed the initial condition. We started with two Gaussians overlapping with each other. The result seems chaotic for both the color-mapped rendering and the histogram. The non-uniform behavior of this example is clear. It seems that the algorithm didn't know where to put the two Gaussians. The curves represent regions that cannot converge quickly. Their effects propagate to other regions and cause the entire result to become uncertain. In fact, the segmentation result in this example was incorrect, as the EM process was trapped to the local maximum. The third example, example C, shows using EM to segment one cell. The cell has a concave shape. We can see that the iterative process is relatively confident. However, the non-uniformity of the process is shown as the noisy patterns in the 2D histogram. If we were only looking at the histogram, we would say this is a highly ambiguous case. Lastly, example D shows the segmentation of a round-shaped cell. We attempted to segment it into two parts. The 2D histogram shows that the algorithm really had some hard time to segment it, as non-uniform patterns can be observed from the beginning to the end. We put the 2D histograms of all the examples together and would like to make sense how we can interpret the results. For qualitative evaluation, we can analyze the uncertainty of a segmentation result by looking at two factors. First, how non-uniform the patterns look like in the histogram. Second, how high the aggregated value achieves. A certain segmentation result should have uniform patterns and low aggregated values. To quantify the two factors, we calculate the mean filtered entropy to measure the non-uniformity, and the aggregation value can be read from the histogram directly. When we plot the values of the examples, we can see that example A is the most certain result, which we should accept. B and D are very uncertain. We can try a different initial condition, so that B may become A. If it still doesn't work, such as D, we can confirm that it is one cell. C is ambiguous and may need further examinations. After looking at the uncertainty footprint in the EM algorithm, now let's look at how the uncertainty footprint works in our graph matching algorithm. As mentioned earlier, we build an interframe graph to represent the spatial relationships of cells. The graph is composed of its vertices and edges. The vertices, shown as colored circles, represent cells. The edges, shown as lines connecting the cells, represent the spatial neighborhood of cells. A problem of applying the uncertainty footprint to the conventional graph matching algorithms is that they are usually recursive and deterministic. As a function domain can be decomposed into a series of overlapping support functions, we decompose a graph into subgraphs. Then, we compute the maximum matching for each subgraph in an iterative manner. Here, for each vertex on the graph, we generate a subgraph by including all edges and vertices that can be visited within a predefined edge length from the center vertex. We update the entire graph in sequence and aggregate the edge changes for the uncertainty footprint. This is an example of applying our modified matching algorithm in the uncertainty footprint to a dataset of zebrafish development. We drew the 2D histogram for the uncertainty footprint as both a grayscale image and a 3D graph. From both representations, we can clearly observe two ridges. According to our descriptions of the uncertainty footprint, the horizontal line represents converged results, and the diagonal line represents oscillations in matching. We can also observe minor diagonal lines of different slopes from the histogram image. These are the tracking issues that we had hoped to detect. We can take a closer look at those tracking issues by isolating and highlighting the cells with many edge changes. Here are several examples. The first example identifies one cell that was segmented as noise in the first time point. The same cell was not able to get matched in the second time point. This conflict kept the cell matching results flipping. The second example identifies an oversegmented cell in the second time point. Although the cell in the first time point was tracked to both parts in the second time point, the result is uncertain. Similarly, the third example shows two cells undersegmented and fused in the first time point. The conflict of matching the two cells in the second time point caused many edge flips. Lastly, we also found a case that one cell disappeared in the second time point. All these examples demonstrate that we can use the uncertainty footprint to detect tracking issues. Since many of the tracking issues are produced by segmentation errors, we can use the previously mentioned segmentation methods to resegment these cases. Now, we can put everything together in our system and make it work. A 4D dataset is first processed with the synthetic Brainbows algorithm. It generates an initial segmentation result. The segmented results are processed with our iterative graph matching algorithm. We detect tracking issues during the matching process, and fix the issues using more sophisticated segmentation algorithms. The uncertainty footprint is used to detect issues and let the segmentation module know where to apply the resegmentation. Then, we detect issues from the resegmentation and allow users to manually correct those issues. Again, the uncertainty footprint is used to evaluate the resegmented cells. Notice that many tracking and segmentation issues are related within a neighborhood. There's usually no need to fix all the issues manually. The partially corrected results are sent back to the graph matching for refinement. The loop in our system is therefore closed. The final tracking results will be refined. We think this is a robust design for our cell tracking system, as different modules can send feedback to each other and guarantee a robust result through redundant checks. We are still working on the improvement of the system. We also plan to include different research groups in developmental biology to try and test our system. Here is a simple comparison between an early development of our system and the tools by Ahmad et al. Ahmad et al.'s methods were published in the journal Nature Methods, and the tools can be downloaded from the website of Genelia Research Campus. They also included a sample dataset of Drosophila development. We applied both their tools and FluoRender to the same dataset. Here is one time point where we can see many issues in the results from Ahmad et al.'s tools. The enlarged region shows that one cell was segmented into two parts. One part was incorrectly linked to a neighboring cell. The correct segmentation and tracking results are shown on the right. With the cell tracking system of FluoRender, we could obtain a 100% tracking accuracy for this dataset. We think that the uncertainty footprint played an important role to achieve it. Here are some notes for the general applications of the uncertainty footprint. The quantification of the uncertainty is indirect. Just like a detective using the footprints to analyze a person's behavior, we are analyzing the non-uniform behavior of iterative algorithms to derive uncertainties. Different applications and algorithms may show different patterns in the histograms generated from the uncertainty footprints. We may also interpret these histograms differently. Intuitively, if the topology of the objective function in an optimization problem is complex, with many saddle points in close proximity, we would expect the path to achieve convergence to be circuitous. Otherwise, the process should look straightforward and confident. For future work, we would like to incorporate more algorithms in our system and use the uncertainty footprint to evaluate their results. We will work with biologist users and organize all the functions in a comprehensive system for tracking different structures from research. The system design will not only be robust, but also provide intuitive operations for routine experiments and high-throughput data. We would like to thank Kristen Kwan for providing the zebrafish eye development data, and Ahmad et al. For providing their cell tracking tool with the example data. This work was funded by the NIHR 01-GM098151-01 and P41GM103545-18. Thank you for attention. You can get more information about our work and FluoRender by visiting the website of the SCI Institute. You can download the source code of FluoRender from GitHub. And finally, you can watch video tutorials on FluoRender's YouTube channel.